Good morning. Good morning. Good morning. Good morning. And welcome to the third of our shared times together to be enthusiastic about all of the invertebrates and the protozoans. So I see that some of you, there's some questions, and that's amazing. I've seen some answers that you've been giving each other in the chat. That's fantastic. I also see some of you finding some of the letters. I'm going to actually erase those circles right now and reveal. And if you're not worried about, you can go back and find them later on when you get the PDF. And we're going to start out today with some reminders. If you haven't already, please scan that QR code and then share with us what your favorite, what phylum your favorite invertebrate belongs to. We'll be using that information later in the year. And the only other kind of housekeeping thing was I noticed that there was an article about blue sky in Science this week. So if you're interested in it, it seems to be a growing place, and I have found it useful. And in fact, some of the material from today came from papers that I learned about on blue sky, which is one of my favorite uses of this kind of social media. All right. Here we go. Now, previously on Zoology 2700, which was asynchronous, so I'm hoping that you found the URL, that the link to the YouTube, to our kind of 45 minutes together, where we talked about these things. We talked about evolution by natural selection, which, like, one of the canonical examples was using invertebrates, the pepper moth. We talked about Linnaean categories, kingdom, phylum, class, order, family, genus, species, which are a vestige of a Linnaean kind of 260 years ago thinking, but we still use a lot of those terms when they are appropriate. We talked about taxa or taxon, monophyly, why monophyly and why tree-based thinking is useful. And then we talked about where do eukaryotes come from? Well, and the serial endosymbiotic theory of kind of accumulated diversity. When you eat something and you don't quite digest it, but then you keep it as a friend forever. So where are we going today? Today we're going to continue talking about eukaryotes. We're going to talk about the sister taxon for eukaryotes, about the timing, just to kind of share make sure that we share the general temporal reference for life on Earth that we're talking about. And then we are going to be jumping into protozoans or protists. And we're going to stay here probably, plus or minus with some digressions, until next Friday. So there's lots of protozoans. Protozoans used to be a course, and it's no longer a single course. So it is a thing that we talk about at the start of the invertebrates because they are really important. And I hope by the end of today, as we talk about particularly parasites and disease, that seems to be a nice entryway, kind of a burr on the human consciousness or on our way of thinking as biologists that gets us in reflecting on why these things are important. And then finally, what are my learning objectives for you, for us, for this shared time together? They are to do these things. So these are from the syllabus, slightly rephrased for some of today's specific taxa. But appraising, synthesizing, and interpreting simple phylogenetic trees. You're going to see more phylogenies, evolutionary patterns, history of protozoans, key morphological innovations of major protozoan taxa, because of course we're not going to do all of them. We're going to do kind of an amuse-bouche of the protozoans, some of the principal lineages, and thinking about all of those evolutionary trends. All right, seatbelts engaged, and the runway is clear. Here we go. So when we talk about the eukaryotes, as we began to on Wednesday in our disjointed way, of course, we're talking about kind of a lot of these are the animals, right? And with the defining features that the cells have a nucleus and a cytoskeleton. And this is the kind of things that get me excited, and I'm imagining they get you excited as well, and that's why you're here with a major that encourages you or requires you to take Zoology 27 and hopefully Zoology 3700. Now, when we're talking about phylogenetics and we're talking about taxa, we're talking about evolutionary trends, of course, we agreed, or I hope I convinced you, or you already arrived convinced to Wednesday's lecture, on the importance of phylogenetic thinking, and one of the ways one of the helpful natures that phylogenetic thinking gives us is a comparative biology. To say comparing to an out group, what taxa, what taxa are in that in group, what characters define that in group, what characters define that taxa. So when we're talking about animals, exemplified here by some of the most numerous in the terrestrial world, the ants, when we're talking about the sister taxon, taxon is the singular, for animals, we're talking about choanoflagellates, which is a single-celled, it's not the ancestor, of course, right? It's an extant thing that we have pictures of today, but it's what we imagine the ancestor of the eukaryotes would look like. So it's the sister taxon that we imagine possessing traits that our most recent ancestor would have looked like. Now, some of those traits involve the fact, and actually some etymology, not just entomology, but etymology in this course. Choan, so choanocyte, so cyte is, say, like a cell, but choan means funnel. And it refers to this shape right here, which you're going to see in a diagram, that there is a flagellum beating inside of basically kind of a collar of microvilli. And that collar is the funnel into which bacteria are captured. So they can live singly, they can live in colonies, they can move, they can look like this, they can look like amoebas, they can move quickly as individuals, they can move very slowly as individuals. But they're bactivorous filter feeders. So this is a single choanoflagellate, and you see the flagellum that beats, that's what the black arrow indicates, and then the blue arrows dashed kind of coming through the purple cone, that indicates the currents that are generated by the beating flagella, and then the little black bits here, those indicate the bacteria that are drawn in on the water current and that are then phagocytized, so eaten, engorged by the choanoflagellate. There's probably more. All of these, that kind of tilde at the start, of course, that's approximately. Very rarely, I'm trying to think of an example where I would say it's likely less than. Almost always that tilde means greater than. And maybe even I'll start to adjust that in some of the slides where I still see the tilde remaining. But the tilde means basically that there's 600 names, and there are more than that in the past. Now it's going to bug me. So there we go. Greater than 600. Now, so eukaryotes, nucleus, cytoskeleton, sister taxon to the eukaryote, the choanoflagellates, the collar, the fonzarelli, there's my age, and the beating flagellum, bactivorous. When did these first animals, multi-eukaryotic animals, start to appear on the Earth? More or less 600 million years ago. And when we talk about life pre that, we're talking about the cryogenean. So that's like algomats, some protists, the things that have formed stromatolites. Especially if you've been to Australia, you might have seen these large kind of fossil remains of these first lives in the cryogenean. When we get into the Edicarian fauna here, that's when we start to see animals for which we still see representative taxa in the oceans of the world today. Like sponges, jellied animals, so not just cnidarians, but also ctenophores, and other fern-shaped sessile creatures. But it's largely not a super mobile world. The explosion of diversity and all of the forms and all of the remaining 34 phyla that we see in the Dunn phylogeny, the eukaryotic, the animal tree of life, they have been with us. They appeared about 450 to 500 million years ago in the here in the Cambrian, in a period called the Cambrian explosion. Now, you've been in Guelph for a while. Maybe you've lived your whole life in and around Guelph or in and around this part of southern Ontario. The red squares indicate, and actually all those colors, the colors indicate limestone that was laid down in these eras. So in the Devonian, the Silurian, the Ordovician 450 to 500, the Cambrian, so that's, and then the Precambrian. So Precambrian is, of course, there's not a lot of life in that rock. But in the sedimentary rock of the Ordovician, the Devonian, and the Silurian, so we're right here in square M2342, that's Guelph. There is lots of life. These were sea floors these many years ago, okay? And so when you're looking at, say, if you're sitting on rocks waiting for friends to arrive to head downtown, or you're sitting outside of a store waiting, or if the store itself is built out over your house, maybe has limestone rock involved in its construction, take a look around. If that limestone is from Guelph, or from the Guelph area, that's Silurian limestone, and you can see representatives a lot of the taxa that we will talk about in this course. So you'll see things like crinoids, and you'll see things like bryozoans, or cephalopods, or brachiopods, or all of these kind of marine taxa. They're still, remnants of them are still here in the ground that you're standing on. And early, it's back in 2020. I took a whole bunch of gigapans of this kind of environment around in each one of those red squares, actually. And so if you're interested in exploring some of these later on, what you can do is to head to some of these links and check out some of the fossils and see how many kind of different taxa, how many phyla you can find, and walk through the ocean floor. That link will take you to a gallery of multiple different places: Silurian, Ordovician, and Devonian. All right, Caramel, who is right over here snoozing now, is here to remind me to check out the chat. And so, yes, will we be going into, I see that, yes. Will the info from the textbook that is not in the lecture slides be tested? So I'll give you a hint about how to use the textbook. If you've got the 3 or the even bigger 4, don't read it cover to cover. Use the outcome, use kind of the characteristic bits at the start of the chapter if we're talking about a taxon. So there's kind of one example. There's a box that defines a taxon there. That's a lot of the key morphological and evolutionary adaptations. That's like this synopsis of the group. And then use the figures that I've pulled out and presented to you as a way to kind of grow out like a fungal mat into the text. Read around. What section did I pull those figures from? Read around in that kind of a section. And yes, and then there's the question that I see answered. The lecture notes are posted after class. There's a reason for that. It is not about organization on my part. It's about a pedagogical choice on my part, and I understand that it's not always something that makes me popular. But that's not really the goal in learning, is it? Often learning is uncomfortable, and I am unpopular. So I'm going to look back later on. I've got Caramel reminding me to take a look down and to examine this. So if you've got some other things in the chat, keep putting them there. And keep helping each other out. That's been fantastic. So protists or protozoans, these are, in this course, we're going to use them as synonyms. They appear in this chapter, in this version, in the third chapter of the third edition of the Brusque et al. Text. The term is about as old as the origin of species, actually. That's the first time that we started to use it. Or earlier terms include kind of cute things like animalcules, the very tiny thing. Now, just like the word invertebrate, protists or protozoans, it's not a monophyletic term. It's one that we've inherited from history, that we agree it's like the Tribiani that I talked about in the last lecture. So it's a term. It's a phylogenetic term. So it's paraphyletic and poly at others, not monophyletic. But it's defined largely by being unicellular and lacking tissue development. So we're not talking about organelles and different tissues. So it's neither a taxonomic nor a systematic grouping. Now, so they're enormously ecologically important as primary producers, as consumers, as vital links in food webs, as alterers of the climate. When you think about plankton in the oceans, a lot of times you're thinking about protists, and we're going to talk about some of those examples later on today. Our own species, if we're going to be like species philic, is that we're greatly affected by protozoans, particularly by parasitic ones, either directly or indirectly. And of course, one of the classic examples, so classic that we're not going to double cover it. We're going to talk about Plasmodium in lab next week. Plasmodium is the needleplastid, of course, behind malaria. So I'm going to make a reference to malaria today, but you're going to dig into malaria next week in the lab. They're characterized by asexual reproduction, so the chromosomes split, and then that can happen with binary fission, multiple fission, budding, but not sex, okay? Asexual reproduction. And traditionally, taxonomically and systematically, we used how they move with cilia or flagella or pseudopods. So cilia like a paramecium, flagella like you just saw in the choanocyte of the choanoflagellate, or the pseudopodic action like the kind of creeping towards you amoeboid. We use those to differentiate groups. However, what we've learned with the advent of molecular techniques that let us indirectly look at who's related to whom and how closely is that those ways of moving are super convergently evolved. And so, and you actually heard me refer to that in the fact that the flagella possessing choanoflagellate in certain life history conditions can assume an amoeboid form, so a pseudopod. So beware of movement as a way of categorizing things. Now, here is a phylogeny. This is from the text. When we talk about the Dunn et al. Phylogeny, we're talking about the phylogeny excluding fungi within this small brown triangle. So the arts and literature from Trivial Pursuit of decades ago, because I'm a very old person, that's what I continue to see. So the Episcodonts, but this large phylogeny containing many, as you see, poly and paraphyletic names, but the uniconts, that's what we're going to get to. The red groups include some better characterized and economically important lineages within the prokaryotes. So they're very divergent. We don't know which ones are ancestral, and they are not monophyletic. And I've already told you that being monophyletic is what we aspire to be because of all of the powers. And so perhaps when I speak about these Tribiani groups, you're going to switch to this kind of Swanson reflection of frustration with the phylogenetic method. So I hope I hope that you will respond to that encouragement by staying with me. There is a value to still using shared terms across generations of biology. There's a value to monophyletic thinking and informed by phylogenetic systematics, but please don't be the Ron and head out. Be Ron and build a canoe, but don't be Ron and throw away your computer into the dumpster. Look at me, updating my popular culture lectures by at least a decade from the 1990s to the 2000s, so becoming as old as you. Protists, protozoans, so small single-celled eukaryotes, the first animals, animalcules. They're mobile, they're heterotrophic and autotrophic, so they eat things, and they can also have plastids and harvest and eat the sun. More than 200,000 species, but there are 215,000 described species, and they have been around for a long time. And you've seen them before, as you've looked at in your training as a biologist in high school and even elementary school, depending on where you went. You've looked at paramecium under the microscope, so you've seen these kinds of organellular structures. They're doing jobs, right? There's a division of labor, and this is one of the key characteristics of animals that becomes even more specialized and large when we start talking about building tissues. But we see lots of division of labor within these these protozoan cells. So we can see digestion with the lysosomes. We can see internal support in the cytoskeleton, one of the key characteristics of the group, that there's flagella and cilia that help with locomotion, and there's contractile vacuoles that kind of deal and excrete byproducts across that cell wall. So the first group that we're going to dive into is over here on the right-hand side, and it's the euglenozoans. And it's generally a well-considered monophyletic group. Now, we're going to only talk about two of the three, the euglenids and the cnidoplastids. The cnidoplastids is the first group. So within the larger monophyletic group of the euglenozoa, the cnidoplastids include a whole bunch of important and really interesting groups. And so they have some heterotrophic forms, some autotrophic forms, and some can switch, which I find as a trait particularly kind of amazing and fantastic. And there's lots of cases when I've been hungry and haven't had the ability to phagocytize my lunch that I wished I could use this shiny pate on the top of my head to be an autotroph, but these guys can. So the flagella is largely used. They have a flagella, and in some groups it is like this. So there is the flagella that is bound to the body with this undulating membrane. Okay, so there's in one group like a euglenozoan, so this is an autotroph. So we've got a chloroplast there. We've got this emergent flagellum that's beating and helping it move. Then we've got the chloroplast, and then this paramylon. The paramylon is a particular characteristic of the group, so it's a carbohydrate storage organ or organelle. So, like I said, here is actually a picture. This isn't a single organism that you're looking at. These are two of them. This is the action of being et that is happening here. So we've got a euglena here that's being eaten by the paranemia. So there's a feeding chamber, a rod organ that gives some stiffness to the end of this predator, and then a leading and a trailing flagella. Clonally reproducing, asexual reproduction, and mostly marine. So there is a paranemia moving through the water. So this is the predator down here. You can see that beating flagella that's moving back and forth, can extrude, can pull back. The rod organ for stiffness involved in prey capture right here. And it's made of really tiny micro bundles, microtubules. And there it is, the leading and the trailing, and the rod organ. So cnidoplastids are slightly different within the euglenozoans. The cnidoplastids have this traditional kind of, you can actually see it here. It's the undulating membrane. So as you look at the undulating form of this video kind of looping through here, what you're looking at is the flagella that has kind of flipped over towards the back. That's it there. There's the flagellum, and there's this undulating membrane. And they're characterized, this large mass of DNA at, let's call it the anterior end, a single mitochondrion, so that's the cnidoplast. So if you were to see them under a slide moving like this, that kind of serpentine motion and the undulating membrane are two characteristics that let you know that that is what you're looking at. Now these are where Plasmodium is a cnidoplastid euglenozoan. All right. I'm showing you, after telling you that we're not going to talk about Plasmodium or malaria that much, I'm showing you this picture because this is a paper that came out this week in Nature that talks about, and so the details are there only for your interest, but it's incredibly exciting to mark this kind of moment. They have, they're announcing the production or the development of a single-shot malarial vaccine with more than 90% effectiveness. That uses what they've called genetically attenuated parasite properties, which means they've deleted specific parasite genes that are critical for development in the liver of the host. So they infect in the same way the sporocytes of the Plasmodium or of the vaccine in this case infect the liver, but when they develop, they die, and that stimulates the immune response of the host, of the person, and that produces T cells and antibodies and eliminates further and future infections. This is incredibly exciting because if you know anything about human history, evolutionary history, contemporary, if you know about if you think or care about equitable distribution of the world, malaria comes into this, and so being able to vaccinate against malaria is going to be incredibly important. Now, the cnidoplastid that we're going to talk about a little bit more detail today is one that's closer to home, kind of. I'll get to what I'm talking about there. So we've had malaria in Canada historically. There were mosquitoes that vectored malaria up until the end of the Second World War, really, in the development of DDT. But we're going to talk about Chagas. Chagas disease, which is a Trypanosoma cruzi, which is vectored by this kissing bug. So if you've traveled in Central or South America, perhaps you've been concerned about it or concerned about these true bugs, these kissing bugs, because kissing or Chagas disease is characterized by fever, swelling, pain, heart failure, death. Tens of thousands of deaths per year in Central and South America, and the long-term cost to infection are into the billions of dollars per year, even with this geographically restricted range. So you can see the characteristic needleplastid form with the undulating membrane here. And here's a closer look at the reduvid, the triatoma, one of the species of triatoma that vectors this particular one. So what we see here are let's start out with number one and say the life cycle, and thinking about the bug taking a blood meal that contains these tripomastigotes. I have to kind of run at that one each time. The tripomastigotes, so that's the characteristic of the adult form. They infect the bug itself, but as they circulate in the human, they're forming and replicating with asexual reproduction in case binary fission to burst out and to stay within the bloodstream and to move about. And so they can reside. There's a symptomatic stage, but this kind of infective stage can be maintained in people for a long period of time. So multiplying, penetrating in the cells, moving about inside of the body, then taking a blood meal, infecting the actual bug itself. So here's the triatome bug stages, and then back into the human. Now, why is this one of the needleplastids that I've chosen to focus on in this course? Partially it has to do with the fact that so this is more than a decade old now, this review paper, but these were epidemiological experts who were asked to focus on emergent diseases that, based on the disease properties and expectations of a changing climate, that we would have to deal more frequently with in Canada. And so they're ranked here. Don't worry about the score. The score just means basically they partitioned out using different analyses ways or reasons why we ought to be more concerned about the emergence or the entrenchment of these diseases. So you can see Chagas, just after Giardia, which we're going to come to in a second, was the second ranked disease, and it's right up there in terms of why we ought to be concerned about it, along with West Nile. Now West Nile's already here. Chagas could exist here, this analysis essentially say, and that its entrenchment here is likely to be facilitated by expected and the changes in temperature and precipitation that we're expecting and already experiencing. And one of the only things that's slowing it down right now is the taxa, the vectorate, the triatoman bugs. They're not yet here. And my guess is, my prediction to you, and you can come back to these notes later on, is that it will be in your lifetime. So Caramel says, Smith, go back and look at the chat. And so Smith is scrolling through the chat. What's the difference between taxonomic and systematic grouping? So taxonomic grouping would be like a Linnaean category. A systematic grouping would be one that is supported by some kind of phylogenetic systematic analyses, some phylogenetic analyses. It's likely monophyletic. Some taxonomic groups are systematic, but not all. What made him unpopular? It was the lectures, the lecture notes. I love the chatting that you're doing back and forth. This is fantastic. Protozoans, and protozoa, yes, we're going to use them as synonyms. Can they be auto? Yes, and that's a great answer as well. Mixing up the trophs. Okay, so there's a question. Well, we need to know all the parts of that complicated phylogeny. I'll remind you of something that I said when we were back together on Monday, and again, I think I said it on Wednesday. If I wanted you to know and memorize all the characteristics, that would be a different class. That's not me. What I'm interested is interpreting, synthesizing, and analyzing. Okay? So all of the evaluations in the lecture part of this course are, if I want you to talk about a phylogeny, I'm going to give you the phylogeny. If I wanted you to think about a distribution of traits, of characters, I'm going to give you those traits or characters. So I offer them to you as a way to become familiar and excited about these taxa, but please don't try and memorize, particularly within the protozoans. Do not try and memorize these things. Where do parasites go if there is switching ability? So I'm going to leave the switching because we're going to talk about that with the Giardia that we're coming to in a second. And the immune system, that's correct. But, yeah, I agree. Parasitic wasps, parasitoids should be more popular. But we come back to our Ron Swanson frustration, the protozoans, and we're going to jump. So from the euglenans to the fornicates. right here, this red triangle. Now, there are a couple of groups there that also affect the axostylatans that can give urinary diseases, sexually transmitted diseases that persist in the urinary tract of humans. That would be one option in some years for me to kind of get you excited about the fornicates. But I'm going to switch. Instead of thinking about sexually transmitted diseases, let's think about Giardia. Now, Giardia is exciting. Actually, throw in the chat if you've had Giardia. Throw in the chat why you're excited about Giardia. But I'm guessing out of a group of nearly 300 of you that 50, maybe more of you have had Giardia, whether it's been diagnosed or not. I'm guessing some of you have had it. So what you're looking at here is the written notes of a letter that Anthony van Leeuwenhoek, who, and I apologize for my Dutch, but Tony, this is the inventor of the microscope, who a lot of his colleagues at the time thought he was an absolute bananas crazy person because he kept describing these tiny living things that were invisible to us except using his newly designed apparatus. And one of the cool things that he did was basically look for these tiny protozoans, these animalcules, in the materials that he had available to him, like water, but also his own diarrheic stool. And so he was the first person to describe what Giardia looked like. So one of the very first animals, tiny protozoans, that was described by our species was this species. So, hooray, looking at your own diarrheic stool for the advancement of science. Giardia is actually kind of beautiful, the organism, the symptoms and not so much. But when we're looking at Giardia, this is one of the characteristics of this group is that the mitochondria has been secondarily lost. So remember we talked about the serial endosymbiotic theory as being one of the reasons we think that the eukaryotes have been successful. In fact, and you're going to see this as a reoccurring thread throughout the year, losing that which describes the group, that character which describes the group, is a common mechanism of evolution. They have a duplicated nuclei. That's what looks like these two eyes here. And they've got these kind of caudal or tail. Think about, it's weird to talk about heads here, but we've got these caudal flagella here. So you're looking at a scanning electron microscope here of a pig intestine, and the pig has been very heavily infected with Giardia. So the Giardia has a much simpler life history stage, but it's living out in the world both as these trophozoites, that's the little teardrop shaped, but also these cysts. The cysts are how it persists, if you want to remember it that way. But the cysts can live in soil for a long period of time. And just like the needleplastid, the Trypanosoma cruzi associated with Chagas, asymptomatic individuals can live for a long period of time and be chronic carriers that some epidemiologists refer to as cyst passers, which feels only passingly polite to me. But it has been a harmful parasite. If you've ever had it, you understand this. But it is also kind of commensal. And so it's both in a sense, and in much of the world, Giardia is omnipresent, but the symptoms are not always there. Now, why is that? Now, it has something to do with the phylogeny that you're looking at here, which I'm going to expand and shift over to the left-hand side of the screen. And what I'd like you to do is take a look at this based on what I'm going to share with you, which is that this is a phylogeny made from Giardia that have been harvested from numerous different organisms. And if you were to look at, and you can see the names of the hosts on the tips of the trees there, put in the chat or editate the screen if you want, a number. How many species of Giardia do you think there are? I can tell you there, at this point, when this paper was written a little while ago, there was one. But how many species of Giardia do you think there likely are? I'm going to take a second and drink my tea. 1,000. You're maybe not far off. More than 2? Yep. Lots. Many. I like that. This is an ecology kind of counting. Ecologists count like none, some, many, too many to count. Yeah. Thank you. This is great. Many. Many, many, many. Lots of puppies and kittens. That's exactly right. Good. So this is kind of amazing. So pat yourself on the head. I think I agree with you. This is fantastic. Now I'm going to move this ahead and find my tools to take away those. Thank you for doing that. But this taxonomic uncertainty continues to affect our understanding of the epidemiology of what has been called only one species, because in fact there are likely many species. At the time this paper was written, 15, 16 years ago, this paper can now drive, there were this many taxa. They talked about, some of the epidemiologists talk about assemblage A being a species, another, so 1, 2, 3, 4, 5, 6, 7, and then some outgroups. Now, I think it's more likely, and what sequencing more and more parts of the world, more and more hosts has shown us, is that there are many, many species. So, Caramel jumps in to remind me to look down, and I think I'm up to date. If I have missed some question there, please kind of remind me, because we're going to keep moving. The protozoans keep us moving. And we're going to jump. So we've been here in the euglenozoans, in the fornicates, and we're going to spend some time. Now we're going to slow down, because in the aviolates, which is a well-characterized group that contains dinoflagellates, apicomplexans, and ciliates, so some of the most iconic groups, we're going to take some time that's going to fill the rest of today, but also going to extend into much of next week. So, writ large, the aviolates are characterized by this system here, where they've got this abutting membranous sacs that support the cell membrane. So not just the cytoskeleton, but these seemingly supportive cellular sacs. Now, the first group that we're going to spend, and the group that we're going to spend the rest of the time with today, are the dinoflagellates. Almost certainly you know about dinoflagellates. You just might not call them dinoflagellates. And we'll talk about why, how you may have been exposed to them. Now, about half of the dinoflagellates are photosynthetic. And so some literature and some press coverings call them algae, but they are not. They are these autotrophic animals, these protozoans. They usually have two flagella. They have these three terms that now I offer to you not as something to memorize, but as something, if you do memorize them, that you can use forevermore in Scrabble. They have a sulcus, a cingulum, and a theca. So you can use them either as Scrabble words or as swear words if you're looking for. A lot of these morphological terms really can do good secondary work as swear words that you can use in polite conversation. Theca, if you've stubbed your toe. You're not saying anything bad. So if you have a side gig as in an elementary kind of an environment or with small children, you can use theca. So this is two different. So there's the stradium and the noctiluca. Noctiluca's down here. We have a long flagella, and then they're essentially grooves in some of the exoskeleton that we've used historically to help characterize them. Now, so important primary producers in marine and freshwater environments. So one of the ways they are important is when their numbers change. And so if you've heard of things called red tides, a red tide, this harmful algal bloom, algae is actually a dinoflagellate. And so a red tide often is affecting parts of Florida. If you're going to Florida in your reading break later on, or if you've lived there or have family there before, you've probably heard about harmful algal blooms. And a lot of these are caused by changes in the community structure of the dinoflagellates in the plankton. So when large amounts of them begin to populate the water column, they can not just they do produce oxygen, but they are producing secondary toxins that the animals that consume them are then killed by. Now, this is a big poisonous problem. So this poisonous algae that Florida often ends up being concerned about from a tourism perspective is caused by Carina brevis, which is a dinoflagellate, and it's native to the Gulf of Mexico. The Gulf of Mexico. It breaks out every year, and it produces a neurotoxin that paralyzes and disorients marine life. But not all dinoflagellates are cause harmful algal blooms. We'll talk about those in a second. And not all of them are red. So instead of talking about red tides, the NOAA in the United States has tried to help people along kind of a learning about becoming more bioliterate, is thinking about them as harmful algal blooms, or HABs. So a red tide is and a harmful algal bloom. They're basically more of a social concept than a scientific or a taxonomic concept, because not all dinoflagellates produce neurotoxins. The Noctiluca, which is one of the two pictures that you saw in a previous slide, in which we're going to talk about one of the amazing things that it does out there in the plankton, does not, for example. And the killing can simply be caused by a reduction in oxygen, rather than the production of neurotoxins. So in this decade-old review now, they talked, about different kinds of algal blooms. So Noctiluca, a different kind of Noctiluca, these dinoflagellates here, they produce water discolorations when their proportions increase out of what is normally experienced particularly in coves or bays, protected waterways. So species that are toxic to non-human taxa, vertebrate taxa, and some invertebrate ones, and particularly important to aquaculture systems, include more of these dinoflagellates. So there's some of the Carinas that we just talked about, Carina brevis. Here's some closely related species, two different species of Carina there. And then, when we get into the potent toxins that can work their way through a food chain that can affect humans, if we begin to start eating the dead things that accumulate on the seashore, as a word of suggestion, don't eat dead things that accumulate on the seashore for a whole bunch of reasons, but this is one of them. Why do they change? Why are they these characteristic outbursts or uncharacteristic outbursts? Well, one of the things, they're caused by a whole bunch of reasons, but one of them, as you might have unpacked already in your thinking as a biologist, is that they are anthropogenically forced changes. That as we change the productivity associated often with agriculture of the water and the streams that are running off of the terrestrial environment and entering protected bays and entering into marine systems, those change the nutrient description of the water, and we can change the temperature, we can change the salinity. All of these factors can change the abiotic living environment that can cause outbreaks of the dinoflagellate taxa that can cause harmful algal blooms. So that's a whole area, an active area of research, of both marine and protozoan research, that if you're interested in these kinds of things, dinoflagellates and harmful algal blooms might be a way for you to think about that. Now, if you like the marine environment and the shoreline and the kind of the ooh factor that goes with seeing glowing, splashing water, that's also dinoflagellates. These are the bioluminescent forms that are associated with Noctiluca and other bioluminescent dinoflagellate taxa. So this Noctiluca, which is, you're looking at it, these are porpoise that are swimming through waters that contain Noctiluca, that they're causing the bioluminescence around the dolphins. And generally, historically, we talked about the function about this bioluminescence not being known. What we're coming to accept is that there likely have to do, it's an adaptation at a larger level than just the individual single dinoflagellate cell. And it's associated with this flashbulb effect, which we think is a startling or acting kind of like a burglar alarm. So it's a flash that caused, so the splashing through the water there that the boots were doing, that's alarming. That's a potential predator, that's a grazer, that's a problem. And so the flash is something, the thinking goes, this is a little hand wavy, attracts a higher level predator that consumes the grazer or the thing that is going to be eating the dinoflagellate. So the dinoflagellate flashes not because of the grazing organism that might be eating it, but to attract a dolphin to eat the thing that is grazing it. So that's dependent, that theory, hand wavy theory, adaptive thinking, is dependent on the population as a whole benefiting. And that's maybe possible or more likely because the reproduction is asexual. And so the individuals in that group are so genetically similar. Now, the last dinoflagellate we're going to talk about for the next 4 or 5 minutes together is one that you've grown up probably calling a zooanthelle. and you know about it. Might be the protozoan that you know most about, or you've been exposed most about, or that you might be indirectly most excited about, because this is the one that lives symbiotically with coral. This is the one that allows coral to be the coral that it is. Remember in the economic arguments about why invertebrates, and we talked about the economic impact of corals and coral reefs to both to our society, to human society in general. So those are dependent on the symbiosis between the coral, the cnidarian, and the aviolate, the Symbiodinium, which is the dinoflagellate lineage that is the Zooanthellae. Okay, this is an exciting group to me. So the dinoflagellate, it's a diverse. There's a largely within this genus. So the Symbiodinium is the diverse genus of Zooanthellae that populate stony corals, but that also can live freely. So there are non-symbiotic, free-living Symbiodiniums. Some of them are facultative. They can choose to live freely in the water column, or they live within, and we'll talk about how they will live within the coral in a second. Some of them are transients. They decide what they want to do. They come and they go. And then some are obligate. Some will likely be vertically transferred along with, as vertically transferred, it's like from parent to offspring by the cnidarians. They can reproduce. They can reproduce sexually. They generally reproduce asexually. And it's the mutualism between these two that creates the framework of that coral ecosystem. Both partners are benefiting. Right? The cnidarian is receiving the metabolic byproducts of the algae, of the dinoflagellate. So it's fixing carbon, and then there's also nitrogen assimilation pathways. The dinoflagellates, in terms, receive housing and protection. They can assimilate byproducts of the metabolism of the cnidarian, so like carbon dioxide and ammonium, and likely protection from grazers. So what does the house of the Symbiodinium look like? Well, see these almost little like eyeballs here? They live in a particular part of the cnidarian's mesoglia called the symbiosome, which is a little pocket that the coral forms for no other reason than to house these dinoflagellates. Now, the housing of the dinoflagellates is, as we talked about with the Noctiluca and also the harmful algal blooms, and how we treat the ocean, how we change the chemistry of the ocean, and the abiotic factors like the ultraviolet radiation that's hitting the ocean surface with reductions and changes in the ozone layer in the 20th century. We change the conditions that the symbiosis between the cnidarian and the dinoflagellate have to live with, and never for the better. And that's this phenomenon that you've been exposed to called coral bleaching, which is the expulsion of the coral. So the coral essentially closes the symbiosome. And that's not for its benefit, there's no benefit accrued. It's kind of a consequence of the changes in the abiotic environment. Increases in temperature, changes or reductions in the water quality, increases in radiation associated historically with changes in the ozone layer. Changes in the tide, changes in the how exposed to the upper elements of the water column or the air that coral are during the tidal functions. They bleach and they expel the dinoflagellates. This is an enormous problem. And we'll talk more about bleaching when we talk about cnidarians kind of next week or the week after. And my last kind of expression of excitement for the dinoflagellate is the fact that when I took this course, we thought there was there was a Zooanthellae that infected all of the corals. And as we sample more and more corals from more and more parts of the world, there's a small map here just of this Caribbean kind of analysis, and as we apply genetic methods to look at their diversity, what we're finding is that there are hundreds and hundreds of species. So the same story as we talked about with the Giardia. Hundreds and hundreds of species where we thought historically there was one. And that's being changed by how well we can get to places, so remotely operated vehicles, underwater vehicles. Scuba was a big part in the mid-20th century. And then PCR and other sequencing that lets us look indirectly at who's related to whom. So, dinoflagellates. Be excited about them. You're dependent upon them, and you're trying to avoid them at the same time. Protozoans, protists, they're kind of amazing for us. Now, This is a summary of where we've been, what's coming up. We're going to talk next week on Monday about the apicomplexan, so another alveolate group, and we're going to drill down in that case into one taxon. We're going to spend the whole time talking about Toxoplasma gondii. Next week, your labs start. What not to forget or what to kind of work on along the way. Remember that the labs start next week, so buy your lab manual, your notebook, and your dissecting kit. And then before you go to your lab next week, make sure that you read the lab 1 section of the lab manual before you get there. Don't be reading it as you're sitting there waiting for the lab to start. And then, of course, the quiz, the first quiz associated with the lectures, is going to open in a couple of hours, and it will remain open until the start of lecture on Monday. And you have, as we talked about in earlier times, all of the periods to break out. You have an hour over that whole weekend to start it whenever you want. I'm going to stop recording. I'm going to encourage you to be good to yourself and good to those around you, and see you on Monday. And once I've stopped recording, which is now,